Hello, dear participants. I think we can start. I am Marta Tseduliak, and recently I became a PhD. I defended my thesis on May 25th, so now I'm PhD in biology, and today I'm going to share with you some insights about my research on ribosomes and tRNA, and we will talk about unraveling the language of life, exploring ribosomes and tRNAs through bioinformatics and genetic engineering. One moment. Yeah, okay. So let's talk about DNA first. One moment. Oh, DNA stores the information about proteins and different types of RNA. It comprised of four different nucleotide types, C, A, T, and G. Here you can see U, and this nucleotide is only found in RNA. And DNA is often found in double helix structures, whereas RNA is usually single-stranded and it contains ribose instead of deoxyribose found in DNA. So here we have RNA and here we have our ribose, and in DNA we will only have H instead of OH as it's represented here. Okay, so this ribose allows RNA to fold up into particular shapes. And this ability allows some RNA molecules to have precise structural and catalytic functions, as in ribosomes, and we will see it later. But let's go back to the DNA, and it specifies the sequence of amino acids in proteins. When the cell needs a particular protein, the nucleotide sequence, oh, Um, yeah. DNA specifies the sequence of amino acids in protein, and when the cell needs this protein, the small part of DNA is transcribed, so it is written in RNA language, and then this small piece of RNA is translated, so the protein is made. And today we will focus on the ribosomes. So who does the reading? Ribosome does the reading. Ribosome is a protein synthesizing macromolecule present in all living cells in all living organisms, and it contains of two subunits, small and large, each containing only two types of molecules: protein and RNA. RNA and a lot of proteins. The bacterial ribosome is smaller than a eukaryotic one, and it is depicted here in gray. So this is a bacterial ribosome. And the majority of the rRNA and ribosomal proteins that constitute the bacterial ribosome are conserved in eukaryotes, and therefore they are considered to form the core of the ribosome. Here you can see the similar core, this gray core of the ribosome. The eukaryotic ribosome, in particular the yeast ribosome, appear to be larger and have additional layer of ribosomal protein and RNA, depicted here in blue. In metazone eukaryotes or in animals, like you and me, these RNA protein layers has increased in size and complexity owing to the presence of additional ribosomal protein and RNA extensions. So our ribosomes is larger. Okay. So main components of the protein biosynthesis machinery, the ribosome is conserved across all domains of life, and we have the same core structure of our ribosomes. So as I said, ribosomes are involved in protein biosynthesis, and they read the letters of mRNA in groups of three, known as codons. So here, three, and they are known as codons. Transfer tRNA, depicted here, transfer amino acids to the ribosome to form a new polypeptide or protein, because proteins, they consist of amino acids. So transfer RNA attached to the messenger RNA through the anticodon codon stem loop. So here we have codon, and on the tRNA we have anticodon, and they form some kind of bonds, and then amino acid is attached. If there is a match, amino acid is incorporated into a newly synthesized protein, and the ribosomes shift in exact step of one codon at a time. So it shifts for 3 nucleotides. And here we have our genetic code table, and here we can find out which amino acid correspond to which codon. For example, here we have UGG codon, and we can see here first letter U, G, and G is tryptophan. Okay. And now more about my research topic. And my research focuses on mutations in the RPSL gene encoding for S12 ribosomal protein. This protein is a part of the coding center of the ribosome that recognizes the geometry of codon-anticodon complex. So why we are interested in S12 proteins? It was reported that some RPSL mutations in antibiotic-producing bacteria, Streptomyces, cause antibiotic overproduction, and it is highly valuable trait for combating the modern threat to humanity, antibiotic resistance. So a few words about Streptomyces. There are soil bacteria with unusually complex life cycle. Their life cycle starts with this germination of spores, then vegetative mycelium is formed, and later serves as a substrate for a new so-called aerial mycelium. And this stage of development usually corresponds with the production of antibiotics. Here. Eventually, the aerial hyphae develops into chain spores. And our model organism, Streptomyces albidoflauus, and we are focused on a conserved region of its S12 protein. Particularly, we are interested in residues 88 and 94. And here you can see a multiple sequence alignment, which uses amino acids sequences of S12 protein from different organisms. You can see some bacteria, mushroom, and animals, and yeast, and fruit fly. And this is the way of lining up biological sequences. It can be DNA, RNA, or proteins to see where they are similar and where they have some differences. Remember, I said that ribosome is a conserved structure, and its primary amino acid sequence also is conserved. On the figure, you can see some regions. They are highlighted in red and orange color, and these are similar parts. For example, these parts are similar in bacteria, and this part is similar in all animals. And overall, S12 proteins are highly similar in all of these organisms, and this is really the rare case. And our region of interest is also very conserved, so it's interesting to study. Okay, and let's move on to the ribosome engineering, which is the formal term for introducing the RPSL mutations. It can be done by selecting for pre-existing RPSL mutations in bacterial culture, then confer resistance to streptomycin. Streptomycin is antibiotic that binds to the ribosome and to the S12 protein. So mutation in S12 protein are common resistant mechanism to streptomyces. And by coincidence, it is historically one of the approaches to isolate improved streptomyces strains. And here you can see some mutants that differ in color. The more intense the color, the more antibiotics the bacteria produce. The names of the strains, for example, K88E and V87K, indicate the position of the mutation in a protein and which amino acid was substituted. For example, it was valine that was substituted with arginine in 87 position. The modern approach The modern approach to ribosome engineering involves generating mutants by genome engineering. So we are replacing our wild type RPSL with mutant version. But I won't go into details about this process today, but it's complicated and results in precise genetic alterations only in RPSL gene. And this approach is more suitable for accurate identification of RPSL-mediated effects on antibiotic biosynthesis, which we are interested in. So we generated 3 mutants, a K88E, a K88R, and R94G, and performed standard tests to assess the effects of RPSL mutation on the bacterial cell. Here you can see strains of genetically engineered Streptomyces albidoflavus mutants and the wild type on the plate. We have no difference here, but if we take a closer look using scanning electron microscopy, we can see differences in appearance of spore chains between these two strains. These two strains have an amino acid substitution at the same position, but with different effects on morphology. The third mutant looks almost normal. Then we tested ability to grow or accumulate biomass, and these two mutants, they grew more slowly. Another difference in our mutants was accumulation of the bacterial hormone-like compound, second messenger CDIGMP, which is significantly lower in our mutant K88R. And this is the first evidence of such RPSL mutation effects, but it's still unclear why and how it happens. So we have a black box, a mechanism by which RPSL mutation acts as antibiotic production enhancer. There are some hypotheses, but all of them can only partly explain our situation. Therefore, we try to solve this mystery by means of modern techniques. And we set up experiments to determine the 3D structure of Streptomyces ribosome. And we used Nobel Prize-winning cryo-electron microscopy technology. At first, a few words about this method. This method allows researchers to rapidly freeze molecules mid-movement. So we have our samples, we put it on a small copper grid, and then we plunge freeze it. It means that we rapidly put it in liquid ethane, and all of the particles, all of the molecules, they are frozen now. Then the sample is bombarded with electrons to capture 1,000 images of molecules, and it is done in cryo-electron microscope. The electrons interact with the atoms of the specimen, casting shadows that create 2D projection images of the sample from multiple random angles. And thousands of images are aligned by specialized software, and in the end, we have the final 3D reconstructed cryo-EM density map. So, for the first time, we saw Streptomyces albidoflavus ribosome, at first using scanning electron microscopy, then using cryo-EM. And more than 9,000 images, 2D images like this, were taken. And ribosomes from these images were extracted and classified. So these are our ribosomes extracted and classified. And finally, we have reconstructed the 3D structure of Streptomyces albidoflavus wild-type ribosome. And we also plan to reconstruct mutant ribosomes to determine whether S12 mutations have a dramatic effect on ribosome structure and whether different RPSL mutations have different effects on ribosome structure or not. As you see, this piece of work is still in progress, and the true mechanism of ribosome engineering are yet to be solved. Okay, and the second part of my talk will be about tRNAs and its modified nucleotides. So I will tell you today about tRNAs, and in all our organisms, tRNAs undergo by far the most numerous and chemically diverse post-transcriptional modifications, and they ensure proper structure of tRNAs and their function. You can see they have this peculiar form. So, what are modification of nucleosides? Modified nucleosides are chemically altered versions of the standard building blocks of RNA of tRNA. So it's just a modified nucleoside, as you can see. For example, here we have methylated uridine, or and I'll mention it later today. So normal uridine have here H, but here we have this stick, and this stick represents the methyl group. So 1 carbon atom and 3 hydrogen atoms. And here we have S2UM, so you can see UM, this stick methyl group, and here instead of one of the oxygens, like this, we have sulfur. So it's like a double modification. Okay. And here are the bunch of enzymes that catalyze nucleoside modifications. So different enzymes for different positions in tRNA. And some modifications are common across tRNAs from the 3 domains of life, while other modifications are unique to specific organisms. And our knowledge of tRNA modification in streptomyces is limited. However, it could be necessary for a better understanding of antibiotic biosynthesis process. So in streptomyces, we know a special tRNA, BLDA. Here you can see a fragment of it. And BLDA is able to translate UUA codons, so UUA codons and BLDA. And these codons are very rare property of CD-rich streptomyces genome. And UUA codons are found only in biosynthetic gene clusters and some other genes that regulate life cycle. Currently, there is almost no information about tRNA modifications in BLDA, so our aim was to determine some of the genes responsible for BLDA modification process. So we aim to determine a TRML protein, which is modifying our ribose, so methylate the ribose, and 2 additional proteins that are involved in this modification, 2-thiouridine formation. And position 34 in tRNA is the most frequently modified, so we would like to know the enzymes that modify this position in streptomyces. Okay, and here we use something called reverse genetics, which is almost like reverse engineering. We are starting with a specific gene of interest, identified using bioinformatics. Then we usually create a loss of function mutant by deleting the gene from the genome. And then we observe changes in the phenotype. If we move from phenotype to the identification of mutated gene, for example, we are talking about forward genetics. But here we are talking about reverse genetics. Okay? And at first, let's talk about methyltransferase gene. And it is a bit difficult example because identification of methyltransferases could be tricky. There are many similar methyltransferases within the cell, and they have complex evolutionary history. And we identified 98 annotated methyltransferases in Streptomyces albidoflavus genome. Then we excluded some methyltransferases involved in known metabolic pathways. And since our gene of interest contains a specific domain, we looked only for genes that have this domain, and this is spout domain. And after this elimination procedure, we came up with a few plausible candidates and generate knockout mutants. And the first and the main candidate was the XNR 5296 gene, and I'll tell you more about it. Okay, so for context, there are two positions of the tRNA where methylated ribose occurs. It is 34th position, as I told you in previous slides, and 32nd position. Methylation in these two positions is performed by two different enzymes. And our protein of interest is similar but not identical to either of them. Okay, in gram-positive bacteria, to which Streptomyces albidoflavus belongs, there is only tRML enzymes. And in gram-negative bacteria, like E. coli, there are tRML and tRMJ. And here you can see two multiple sequence alignments, and we can clearly see that our protein, and this is the first protein here and here, have N-terminal domain unlike any other protein. So this, like the very beginning of the protein, is unique, and no other tRNJ or tRML have this domain. However, at this stage, we still don't know if our protein is tRML or tRNJ, because it's a bit similar, but we don't know who is who. However, we aim to identify tRML since there are no data on tRNJs in Streptomyces, and we thought that this protein is tRML. Okay, let's move on. And the next our step was to perform a structural comparison of the proteins. And we generated 3D structure of 5296 protein, our protein of interest, using AlphaFold, and superposed it with experimentally obtained structures of tRMJ and tRML. So here we have superposition with tRMJ, and our protein of interest is purple, and tRMJ is blue, and here purple is our protein, and tRML is orange. As you can see, there is an obvious region in our protein that is absent in tRMJ and tRML, and also this part of the molecule is not ideally aligned with our model. Therefore, we cannot clearly confirm that which type of protein is our 5296 protein. Okay, and the gold standard for identifying gene function is generating a knockout strain, which is what we did. And we were interested in tRNA modification analysis, so we isolated tRNA from Streptomyces albidoflavus strains and performed two types of analysis. The first one, LC-MS analysis, helps us answer the question, which modifications are present in the sample? And during LC-MS analysis, we need to cut our tRNA into nucleosides and separate them by chemical properties, and information about the position of modification will be completely lost. But the second method is specific sequencing method, and it is used to detect only methylated ribose. So it won't give us any information on any other modification. And we combined these two methods. And what did we find? First, we identified a significantly reduced amount of So here is wild type strain, and this is a peak representing modification. And in the mutant strain, you can see only a small fraction of this peak. Okay, and the second, the position of that was not and position of that that disappeared was not in BLDA as we expected, but it was in different tRNA species with the codon GAG. And here we have unmodified U in 5296 knockout mutant. So our gene of protein of interest is tRMJ, not tRML, and it is the first demonstration of tRMJ-controlled methylation in gram-positive bacteria, and it is a quite unexpected discovery. And this is an interesting new fact in streptomyces biology. But the main question about tRNA enzyme remains unanswered. Okay, and the next short story will be about two enzymes, MNMA and YRVO, involved in S2UM formation. So here we have our UM that we are familiar, and these two enzymes catalyze the formation of S2UM. So here we have UM plus sulfur instead of oxygen in this position. It is a second position in this base. So bioinformatic analysis here was much simpler because these enzymes are very conserved and their genes are often located nearby. We identified such genes in Streptomyces albidoflavus, and you can see it here. That in Bacillus subtilis and Mycobacterium tuberculosis, these two genes are also situated like this very closely. And we have a similar picture in Streptomyces albidoflavus. So we generated knockout mutants for these genes. Okay, and I'll show you some bioinformatic analysis first. In the case of MNMA enzyme, our candidate was XNR 1345, and it showed high similarity on the multiple sequence alignment and almost perfect superposition with E. coli MNMA. And similar picture we can see for the 1347, which is YRVO, and we have pretty good multiple sequence alignment, and there are highlighted some most important residues for catalytic function, as well as here we have some conserved catalytic residues, and they are present in both of our proteins. Okay, and you need to remember that bioinformatics is a powerful tool, but it only gives us hypotheses to test. So we analyze the tRNA modifications in knockout strains, and LCMS analysis revealed the absence of S2UM modification in the strain lacking 1345 gene. So we can, we can see in a wild type peak here, but no peak in a mutant strain. And we can confidently say that this enzyme, this gene encodes MNMA enzyme involved in S2U biosynthesis. But in the case of 1347 gene, we did not observe any alteration in the tRNA modification spectrum. So we have the same peaks here. And therefore, we conclude that 1347 is not involved in the tRNA modification process, despite bioinformatic evidence of similarity to known VIRVO enzymes. And in conclusion, I want to end with the words of famous microbiologist Jacob Monod. What is true for E. coli is true for elephant, and fundamental biochemical and genetic rules of life are universal across all organisms. And since all living things share a common evolutionary ancestor, biological mechanisms first discovered in bacteria will have similar mechanisms and basics for other organisms. And we have much more in common than we think. And don't underestimate bacterial research. They can and already have helped us so much in understanding the basis of molecular genetics. And if you would like to learn more about molecular biology and genetics, register for our course, and you will be also able to independently design and implement some pipelines for analyzing biological data. So thank you for your attention, and I am ready for your questions.
So thank you, Marta. And I believe that we will have questions in audience considering the talk itself and some questions perhaps about the program. So I would invite those with the questions about the talk, and then we can answer some queries about the program. So please, guys, go ahead. So you can either raise hands or you can just talk directly. So no pressure.
Hello,
Hello.
my name is Julia. Thank you for lecture. It was really interesting. And I want to ask about a language program because this lecture, to be honest, it was difficult for me to understand. I didn't understand anything, and I want to ask about this in program. Is it will be all the time in English, or sometimes we will have in Ukrainian?
Yeah, so Yula, thanks. Uh, we've decided to switch into English this year because we've observed that people are generally able to comprehend, and although it might sound or look frightening at the beginning, but then even people with maybe lower knowledge are starting to adapt and tune in. But generally, if you are not sure about your level, so I think that we've estimated the necessary level as B2. So if you don't have B2, you should be probably preparing for a hard ride because although inter-student communication and communication on practical lessons like labs or maybe some bioinformatics practicals will be partially at least in Ukrainian, but the lectures would be in English. So yeah, B2 is your threshold, I would say.
Okay, thank you.
Okay, thank you, Yula. Okay, so we had question about the program before the questions about the talk. So I guess we can mix up these two categories. So please go ahead. Yeah, you can ask in Ukrainian if you want as well. Okay, guys, we will still remain online. I will check some questions that were perhaps pre written before the talk. So there was a question from Kim Nayon. Do we
Mhm.
have him? No, we don't have him online. Okay. Then there was a question from Sink Gurte. Do we have Sink online as well? Okay, I
Mm.
guess we can answer them in case they are going to watch this in a record. So, Marta, question from Sink Gurte. For how many years were you researching in this field?
Mm. I have been researching in this field since I was 19, so it's like 8 years now. And I have like 4 or 5 years experience of mentoring the students and 8 years of experience in lab work.
Okay. So, right. So, about the question from Nayon, I guess he's actually uh, he studies in Ukrainian Catholic University, although he is definitely Korean. So he says that he works in a sphere of language education. So probably about it goes about linguistics, but he's very interested in science and he would like to join, and he likes to listen in Ukrainian. So I guess yeah. It's a bit disappointing for him as well that we are going to use English throughout the course. Anyhow, he also asks that he read a talk recently, and the talk was about the fact that the cells could be reprogrammed using ribosomes as switches ons and offs. And if it's really possible, how would you program your cells with ribosomes? So the name of this technology developed by research team POSSEC from Korea is the RATEX platform, and the result of the study were published in the latest issue of the Angewandte Chemie. Okay, let's check what's a RATEX platform, and then maybe I will give you more. Yeah, so this RATEX, reading the abstract from the paper.
Yeah, I have seen this abstract, and it's something about
Okay.
synthetic biology and ribosomes. And they fused some RNAs to ribosomes and therefore targeted ribosomes to switch on or switch off translation of some genes. So it's a bit like difficult to understand, and probably I'll read about this more later. But it's interesting technique, and maybe we can use it to enhance antibiotic production. Who knows?
Okay, so and then the question is, which genes would you turn off with RATEX?
Maybe I would like to turn on some genes, some regulators, positive regulators of antibiotic biosynthesis.
Okay. Okay. So guys, we had two questions from the registration form. Still, some people online. Do you have any questions? Otherwise, we will wait for additional couple of minutes and then we are going to close the session.
I have another question. And
Uh,
you say I can speak in Ukrainian, yes?
Okay, Юля, please go ahead. Без проблем.
Хотіла задати стосовно практичних і лабораторних робіт, тому що я якби випускниця державного університету, і в нас там, якщо вони проводились, то тільки там в режимі онлайн. Так як я дивилася в соцмережах, то я бачила, що там є якісь такі у вас більше можливостей. Але водночас форма навчання там вказано, що онлайн. Я щось не дуже зрозуміла, коли вони будуть відбуватися, і які там саме будуть види лабораторних і практичних.
Так. Отже, Юля, дивіться, формат, який був цього року, він був такий, що в нас є два практичних курси. Тобто це, ну, якщо так говорити не фенсі назвами, а простими термінами, один курс це прикладна біоінформатика, а другий курс це генетична інженерія. І ми мали обидва курси в другому семестрі, тобто зараз, і маємо їх досі. І в нас, так як багато людей з Києва чи з Вінниці, з інших міст, то ми проводили ці заняття по суботам, так, щоб один тиждень були лекції, а другий тиждень лабораторні практичні. Тобто їм було достатньо приїхати раз на 2 тижні в суботу. Але це все одно цей формат нам не до кінця сподобався, тому що там були проблеми з квитками, були проблеми з плануванням, з обстрілами, з усім. Ну, тобто, ви розумієте. І Цього року ми придумали інший формат, тобто ми з ними радились, отримували від них фідбек. І ми придумали таке, що оці два наших практичних курси будуть в другому семестрі, як і минулого року. Але лютий, березень і квітень це будуть 3 місяці інтенсивних лекційних занять, так, тобто теоретичних. Ми прописали це так, що якщо ти резидент України і львів'янин, то ми би хотіли, щоб ти приходив на кампус офлайн. Так? Ну, якби ми бажаємо цього. Але, ну, звісно, якщо є робота, все решта, то тоді ти можеш слухати лекції в записі і просто виконувати домашні завдання, ну і тоді це буде нормально. Якщо ти не резидент Львова, але працюєш в Україні, то тоді теж ми наполягаємо на те, щоб ти хоча б з'являвся в Zoom, так? Тобто, щоб лекція була, коли лекція йде онлайн-трансляція, будь ласка, під'єднуйся, слухай. І, звісно, якщо ти за кордоном і таке, то різниці в часових поясах ти можеш приєднуватись в Zoom, можеш слухати в записі і так далі. Але травень і червень це будуть 2 практичних місяця. Тобто весь травень, кожної суботи, там буде по 4 пари підряд. Це будуть лабораторні роботи з генетичної інженерії. Якщо ви питаєте, що там буде повний цикл від виділення ДНК і до створення трансгенних бактерій. Тобто ми вивчаємо все, починаючи від виділення геномної, плазмідної ДНК, роботи з ДНК в гель-електрофорезах, ПЛР, різні види ПЛР, клонування і так далі, так далі, так далі. Тобто це прям... В нас дуже класно ці практичні повиходили, в нас є вже база і слухачам подобалось. Тобто ідея така, що ви прослухали лекції, який міні-контрольну, яка дає вам змогу, як дозвіл до цієї лабораторної. І тоді ви чітко знаєте, що в травні кожної суботи вам треба приїжджати до Львова на наші лабораторні роботи. Це означає, що ви можете планувати фактично за рік, хоч зараз, які завгодно відпустки, відгули, замовляти квитки на потяги і так далі. Тобто це не так, як цього року, коли волатильний графік, вам треба кожного другий тиждень приїжджати. Ні, це все ми збираємо разом, компресуємо в один такий робочий інтенсивний місяць. І це буде ефективніше також в контексті навчання, бо ми були змушені скіпати деякі експерименти, бо вони там тривають більше, ніж 2 пари. Так? Чи, наприклад, ти проводиш експеримент, через 2 тижні ти аналізуєш результати, це вже такий розрив в знаннях, студенти встигають забути, це вже для них не так актуально. Тобто, ось на цей травень ми прям вимагаємо, щоб всі були присутні. Тобто, тому що, якщо ти не будеш працювати руками, то цей курс не потрібен. Те саме в червні для біоінформатики. Тобто, теж червень, 4 пари, кожної суботи приїжджаємо, виконуємо повноцінний проект, починаючи від збору послідовностей літератури з баз даних і закінчуючи різними рівнями біоінформатичного аналізу цих даних. І залишається також в нас ще і липень, але там будуть теж семінари, якісь підсумки, тому ми, в принципі, теж дозволяємо онлайн відвідування. Ви не зможете доповісти на семінарі в записі, правда? Тобто вам треба бути в Zoom. І, мабуть, ми будемо просити всіх приїхати ще наприкінці програми для підведення підсумків, вручення сертифікатів, якоїсь там піци, ну, якоїсь такої вечірки, щось в такому контексті. Тобто, ще раз, підсумовуючи те, що ви в нас запитали, перший семестр це будуть виключно лекції, семінари, тому що це, ну, якби начитка, так, теоретичний курс. Другий семестр два практичних курси, де перші 3 місяці будуть іти теоретичні лекції, потім буде травень інтенсив з генної інженерії, червень інтенсив з біоінформатики. Ну, мені здається, що такий формат набагато більш accessible для людей за меж Львова, аніж те, що ми практикували цього семестру. І ми подивимося, як він, як то кажуть, plays out. Можливо, він вже і стане усталеним на всі наступні роки. Можливо, ми будемо його далі якось міняти і адаптовувати.
Стосовно практичних на другий модуль, там де по біоінформатиці, ви казали, що буде там різний інформатичний аналіз даних.
Так.
То ти маєш на увазі, треба знати якісь мови програмування, чи
Ну,
там.
так, дякую. Тобто, знову ж таки, я наголошую на тому, що 95% наших робіт, які ми будемо проводити, це будуть або веб-застосунки, або застосунки з графічним юзер-інтерфейсом. Тобто, ну, знову ж таки, в нас були цього року люди, які взагалі, ну, не володіють ніякими мовами програмування, і тому було прийнято рішення іти таким способом, що ми розказуємо теорію, ми показуємо вихідні джерела коду для тих, хто може його застосовувати, але для тих, хто не може, ми працюємо з онлайн-застосунками і застосунками з графічним юзер-інтерфейсом, тому що наш курс називається «Основи прикладної інформатики». Так? Ми хочемо дати оглядову картину, а вже тоді ці люди, вони можуть далі якось розвиватися. Бо в нас є там кейси, наприклад, коли курс називається біоінформатика, і там півроку читають прям тільки аналіз даних RNA-seq. Ну, тобто там фактично читають тільки транскриптоміку. Так? Ну, ми не хочемо йти в якусь суперспеціалізовану царину, тобто ми хочемо дати основу, так, яку будь-хто, хто має комп'ютер, може застосувати. А вже потім, якщо в нас є в пулі люди, які ну, мають навики роботи там, з мовами програмування, там, з R чи з Python, бо майже всі біозастосунки вони або на Python, або на R, ну, вони можуть далі розвиватись якось. Тобто вам не потрібно знання мови програмування, але ми не забороняємо їх використовувати.
Дуже дякую. Я почула, що хотіла почути.
Так, дякую. Okay, so now we have a question from Daria Navrotska. I thank you for an interesting talk. Could you please clarify what are the working perspectives after finishing course in bioinformatics, for example? So again, you are not applying for your bachelor course or your master's course. You're entering a limited 3-course certificate program. And you receive certificates and credits for this program. So this means that you can, for example, resubmit these credits in any other teaching program or learning program that goes in frame of Bologna process. But it doesn't mean that you receive a diploma of Master of Science or whatsoever, that you're a bioinformatician, because you have like only one limited course. So, what are your working perspectives? Well, I guess if you would like to apply for a position in some med genetics testing lab, and you already have some diploma, master's or bachelor's in biology, but you do not have bioinformatics there, then you can provide our certificate showing that you are at least familiar with nucleic amino acid sequence analysis tools, which may give you some advantages before your peers. But again, so we had this question previously. So it's not a master's course and it's not a bachelor's course. We are not giving you a diploma in bioinformatics or in gene engineering and molecular biology and whatsoever, because your bachelor's or master's course is years of studying. A lot of various courses, preparation of your thesis, etc. So, am I able to give you an answer, Daria, or you need some more clarifications? Um,
No, thank you very much. It was exactly enough. Thanks.
okay, Daria. So finally, we are talking, right? So, do we have any more questions? Please go ahead in Ukrainian or in English. Mm. At least I don't hear the crickets. I guess it's too rainy for the crickets in Lviv. Okay, so then let's wrap it up. Thank you, Marta, for your talk. So, Marta is maybe one of, I don't know, 10 or 20 people in Ukraine that actually seen and worked with cryo-M. We don't have it in Ukraine, of course, and I believe that we have it only several in Europe. So, do you have any information? Maybe in Germany, I don't know, in UK probably.
I just know that the nearest cryo-electron microscope to us is in Poland.
Okay, so
Yeah.
if Poland has it, it's not that rare, I
Yeah.
guess.
Yeah.
Okay, so thank you very much for visiting this talk. And yeah, if you like what are we trying to achieve, go ahead, register, and be advised that we are going to give some additional webinars. So next time, in 2 weeks, I will probably be talking about another, maybe more practical topic. I will give a talk about antibiotic resistance and how does practical gene engineering and bioinformatics help us to understand this unfortunate phenomenon. So, thank you very much, and see you either on a next webinar or on our course next teaching year.
Yeah. Thank you. See you.